welcome to the makeup discussion on Chapter 6. I'm going to pick up where we left off last time on this section. And so we're going to be talking about microbial nutrition and growth. And in particular, we're going to talk about oxygen and how toxic oxygen is to living systems, in particular, of course, the bacteria. So the superoxide ion, which is the natural form of oxygen bonding to itself, because it needs the additional satisfaction of having all of its covalent bonds made. In this case, it'd be double bonded to O2. So, and it takes over a negative overall charge. And then, of course, hydrogen peroxide, which we use in the laboratory, has similar characteristics. It's highly reactive, and then the hydroxyl radical itself, the OH. So these are the topics that we're going to be talking about. Most cells, of course, have enzymes that deal with these oxygen radicals, superoxide dismutase in particular, and catalase, which we've seen in the laboratory. Superoxide ion is converted into hydrogen peroxide by superoxide dismutase, and then that's converted further into harmless water by the enzyme catalase, as you can see here. And so the catalase, you can see the chemical reaction, the O2, the oxygen radical being handled by superoxide dismutase into hydrogen peroxide, and then catalase overall converts that into just water. So the other term for catalase, I guess, encompassing both superoxide dismutase and that peroxidase activity, turning it to water, as you can see here. And that's the overall configuration. Now, keep in mind, the reason that we worry about oxygen and respiration is the presence or absence of oxygen, and which changes which way things are metabolized. So in this case, as we know, we have to drive the ATP syntases in the mitochondria, basically charging that battery through that double membrane, across that double membrane actually, forming that battery effect, and then turning the turbines for the ATP syntases, and then of course that forms all of the ATP that we need, or the bacteria need for their cell. So aerobes, of course, use that gaseous oxygen in their environment. These cannot grow without it, and these of course include most of the fungi, protozoa, and many different types of bacteria, such as the bacillus, mycobacterium tuberculosis. Well, it's a respiratory disease, and it's not shocking to find that a respiratory pathogen would need lots of oxygen. That's why they kind of choose that portal for their infectivity. So anyhow, so the oxygen utilization, you can see the various ways that we can represent that in a tube. You can see the aerobic, microaerophilic, facultative anaerobics, aerotolerant anaerobics, and the anaerobics overall. You can see where in the tube they grow, and that's due to the concentration of the oxygen, as you can see across here, high going down to low. Of course, the oxygen being at full atmospheric oxygen at the top, diffusing down through the tube. So I just wanted to kind of represent the O2 tension that way, and then talking about the aerobic, of course, they would favor the upper part of the tube, so in the growth. So it's easy to see where that upper growth is. It's much darker in terms of growth in that growth medium. Aerobes also can be microaerobes, we call microaerophiles, and they're harmed by normal atmospheric concentration but require small amounts of it for the metabolism. And Helicobacter pylori and Borrelia burgdorferi, which is a causative agent for Lyme disease, fit into this category. And so you might see the movement downward. Let's go and look down from the surface. It grows very readily there, and it chooses, of course, the oxygen tension it can handle, and that's where we see the growth of that organism. And of course, that's due to the slightly lower tension of the oxygen as it permeates into the gases permeate into the tube. Facultative anaerobes basically are adapted for really kind of any environment. You can see the growth is pretty much across the tube, and you can see that represented all the way from the top to the bottom. And the only one that does that is facultative, so you can kind of readily see where that might be. Anaerobes lack the metabolic enzymes for using oxygen at all. Obligate anaerobes lack the enzymes for processing toxic oxygen. As a result, they'll die. And in this group, you can see that they require, in the absence of oxygen for its type of fermentation, leading to formation of lactic acid, and it's not as productive in terms of making energy. You can see the anaerobic at the very farthest away, which would have the lowest oxygen tension down towards the bottom of the tube. So air-tolerant anaerobes do not utilize oxygen but can survive and grow in the presence of it. This is important. Certain lactobacilli, streptococci, and Clostridial species fit the bill for that. And you can see the air-tolerant in this system here in that particular tube. And it grows wherever it can adapt to the oxygen. And even though it doesn't need it, it can tolerate it. Capnophiles, now on the other side of the spectrum, utilizing carbon dioxide. These organisms that grow best at higher CO2 tensions than normally present. And these organisms, Neisseria gonorrhea, Neisseria meningitidis, Brucella for undulant fever, and Streptococcus pneumoniae are examples of capnophiles. pH. Now, pH is one of those things that bacteria can't really become resistant to. They require, the pH to be within a certain range. And if you think of it, that's because of the enzymes. If the enzymes are exposed to various different types or levels of pH, they change their shape. And structure and function. And if they change their shape, they may not function. And as a result, the organism can't survive. So if you just remember, pH is percent hydrogen. As we see here, percent hydrogen is really just the amount. If we consider an acid, we consider the concentration of the hydrogen aspect or the positive proton to be at 100% at 0, where the OH, the other portion of the water molecule, is at the other end of the scale is 0%, where at 14 would be the hydrogen proton concentration is 0, but the OH concentration of the hydroxyl group would be 100% at that end. So they are inverse proportional from one end to the other. So increasing hydrogen ions in the reds showing here, and increasing the hydrogen or the proton at the other end. So 100% at this end for proton, and 0 at the other end for the proton, versus 0 here for the hydroxyl, and 100% at the other scale. So we've already talked about the pH. So what type of organisms, the acidophiles, can survive in the acidic environments? Euglena mutabilis, which grows acid pools between 0 and 1, which is amazing. Thermoplasma grows in the coal piles at pH 1.1 to 2, and Picrophilus thrives at a pH about 0.7. Molds and yeast tolerate acids and common spoilage, of course. We see that. Alkalophiles are at the other end. Natromonas lives in hot pools and soils up to pH of 12, which is amazing. Proteus creates an alkaline condition to neutralize urine and colonize and affect the urinary system, like Proteus mirabilis and those types of organisms we see as pathogens. Osmophiles live in habitats with high solute concentration. So we're talking about osmotic pressure. So the osmo gives you that sort of implication. Halophiles prefer to high concentrations of salt, and halo indicating that being in the high concentration of salt. Obligate halophiles like Halobacterium halococcus grow. We typically 25% salt. Facultative halophiles resistant to salt, even though they normally don't resolve in those. And Staph aureus can grow on sodium chloride media ranging from 0.1 to 20%, which is quite amazing, as we've seen Staph doing that in the lab. Now, radiation-wise, phototrophs use visible light rays as an energy source. Non-photosynthetic microbes tend to be damaged by toxic oxygen products produced by the contact with light. Some microbial species produce yellow carotenoid pigments to absorb and dismantle toxic oxygen. And then, of course, UV and ionizing radiation can be used in microbial control, which we saw in the laboratory. Pressure, for those scuba divers, bariometric type of parameters talk about the weight of the water, essentially, or the pressure. Exists under pressures ranging from a few times to 1,000 times the pressure of the atmosphere. So 1 atmosphere is about 4 feet of going underwater. These bacteria are so strictly adapted to high pressures that they'll rupture when exposed to normal. So the barometric pressures are quite an interesting characteristic, but we don't worry too much about that for the pathogens. There are other rare environments that microbes can grow, of course, and you could just talk about various aspects, but we're not going to go into it. It's not relevant for the pathogens that we study. Symbiosis is a term talking about partnerships. It denotes a situation in which two organisms can live together. Mutualism, organisms live in an obligatory but mutually beneficial relationship. Commensals receives benefits while its partner is not harmed or benefited. And parasite, of course, is a win-lose scenario. So mutual is a win-win. Commensalism is a win for one, and it doesn't really change the other. And parasite is a win-lose. Antagonism is an association between free-living species that arise when members of the community compete. Antibiosis is the production of inhibitory compounds such as antibiotics and surrounding that destroy other microbes. So these are ones that tend to be little pigs, and they exclude others from growing anywhere near that the environment. Common in soil or mixed communities where everything is about competing for various resources and space. Synergistic or synergism is the interrelationship between two that benefit not necessarily for survival. We talked about this in biofilms, but together, individually, they produce at a certain level, but together they go beyond what either one could do. Gum disease, dental caries, various things are synergism. And so synergisms, of course, are biofilms. So a pioneer organizer initially attaches to a surface, other microbes then attach to the bacteria or polymeric sugars or protein secreted by the colonizers. So we see this as well. And just to refresh your memory, we get the initial organisms growing at a particular site. We don't brush our teeth. And then more bacteria come in, and we start to see the biofilm forming, and then next thing you know, you've got a whole bunch of stuff that needs to be cleaned off your teeth, and that's sort of the biofilm. And again, synergistic reactions with multiple different types of microorganisms. Different genes are expressed from these organisms. Okay, now we're going to shift gears and talk about the growth and comparing growth. Of course, just remember that the bacteria don't use any sort of sexual type of replication. They use binary fission. They just, one cell, boom, splits into two. It duplicates its chromosome and then divides between those two chromosomes, and so all the materials inside the cell are equally proportioned. As you can see, here's one cell. They start to divide and split the materials, and then ultimately now you get the two cells. And there's not really any surprise other than the fact that how fast they can do it. And so a generation time is that time required to complete that process, and on average it's about 20 minutes or so, which you think about doubling every 20 minutes, that's pretty scary. So your book refers to as 30 to 60 minutes, but on a general rule I've heard, or I've always been taught, it's about 20 minutes. Shortest generation times are 10 to 12 minutes. Mycobacterium leprae generation time of 10 to 30 days. So it tends to take its time a little bit there, as you can see. So however long that period of time that it takes, we call that the generation time. Most pathogens have relatively short generation times. If you think about it, they got to do what they do, get it over with, and get it out. And so we can look at the math involved in a double. So we refer to this calculation as 2 factorial because, of course, it's the interactions of just one splitting into 2. We look at that as a factorial 2. And so the exponent would be the number of the divisions or the generation times. And as you can see, it can grow quite rapidly. So 2 to the 4th, or having 4 times at which it generates its daughter cells, we see 16, and then that doubles to 32, and then on and on. But you can see either logarithmically, or if you look at the asymptotic relationship, it goes straight up. And so that's why we like to represent things in nice logs, because this is kind of hard to measure if you look at just the flat numbers. Again, it's 2 raised to the power n, where n is the number of generations. So 2 times 2 times 2 times 2, in this case, is 16. If it has another generation, it would be 32, and then so on and so forth. Doesn't take long for this to generate. This equation shows the same thing, except adding the initial number of cells at the very beginning. And so you can calculate that pretty easy. That's the generation number. So the growth curve is a predictable pattern. The growth, we can see if we were to inoculate, let's say, broth that has sterile broth that has good nutrients in it, the organism will start to grow. And we can take samples during that growth, and we can actually count the numbers. After 60 minutes, 120, 180, you can see the sort of the pattern as we grow across. And it's just a simple a series of straight measurements as we do that. And then we would plot that data either in a linear or a logarithmic, which logarithmic is preferred because you can see that this linear approach is going to change to where it's almost useless, where in the logarithmic we can make measurements and from a plot and derive some information. And that's used most of the time. So you can see that we can use the logarithmic scale and looking at absorbance using a spectrophotometer, which is once you've done a standard and you've actually done the plating, you can rely now strictly on the absorbance, and you can see by doing that it makes life a lot easier, and you can actually come up with a number. Again, the linear scale is not too useful for that. And so we can look at, on a logarithmic scale, the numbers. So that would be 1.2, if you look at roughly where it is on the logarithmic, times 10 to the 11th. You can see the numbering system. And you can also plot it as a function of absorbance on a logarithmic scale, which works very nicely. So the different stages of the growth, the initial phase is called the lag phase. It takes a while for the organisms to kind of get geared up for their metabolism and all their enzymes and stuff, and we refer to that as sort of the lag phase. And there's some interesting dynamics. I won't go into that now, but pharmaceutical companies worry about, how to scale these sorts of things. The exponential growth is the actual log phase or the logarithmic phase, and that's where the growth occurs geometrically. In other words, we're wide open. These are multiplying just as fast as possible, ultimately coming into what they refer to as a stationary phase, where the cell birth and death rates are equal. And then eventually we start to decline, where cells begin to die at an exponential rate due to the buildup of waste and other aspects of this. So that's the death phase. And then eventually we get to the point where we can't even measure viable cells. So here it is in a graphic form, and lag, and then we go into the exponential growth, and then kind of come into the stationary phase, and then eventually the waste products tend to cause death, and we now lose the number of cells and no longer maintain. So that's a standard growth curve. The reason that we see this is a tendency for populations to exhibit these sorts of things. We want to measure them for various environments, and so we can make predictions as to what, how these are growing and that sort of thing, or how they might spread. So we use the growth curves to understand the organism's characteristics. Without actually culturing, you need to culture for the calibration, and then once you've calibrated it, as you measure it towards turbidity, in other words, how much the cells actually block the light, and so we call that a turbid or cloudy as the microbes grow in it, and it is a one-to-one relationship if we plot it to the initial plating out of microorganisms, and we can now relate the density to the numbers of bacteria, which really works very well. You can see the 100%, we don't see any opacity here, where now you can see how turbid that culture is very high, and that would block a lot of the light, so you would see that the amount of light being transmitted is very low compared to the 100% in the initial sample on the other side. So you can see, as a result of the transition all the way through, if you were to take plots as it was growing. Very easy way to do it instead of having to plate, and it gives us the data that we need. The other ways of doing this is that we can use counting using a direct cell count measured microscopically. We call it a Coulter Counter, which is electronically scans as a fluid. In other words, every time a bacteria cell blocks the little laser light, we get a count. Flow cytometer works similar, but it measures cells and differentiates between live and dead cells, and using a flow cytometer. And then, of course, we can probe with PCR to quantify just the presence genetically that the microorganisms are there. We can do direct microscopic counts with a grid system, so that way you can measure. We know how many grids that appear on a particular sample. All we have to do is just count one area and then multiply that by the numbers on that particular grid, and we can come up with our calculation. So there's a Coulter Counter, a picture of one there, and it looks at the change in impedance as things flow through it. So we have sort of a magnetic flow, and if something disrupts that, it's sort of like a proximity sensor. It measures that electrically. Flow cytometer, you can cell by cell, you can even tell if the cells are viable or not based on the integrity of the cell itself, and so you can see how they work. And then the real-time PCR, we're going to amplify, and this is just a genetic looking at the presence or absence of that particular microorganism, and then looking at how much of that DNA we can copy at any one particular time. But we're not going to concern ourselves too much with that. So if we were to ask a question like this on an exam, put the steps of these growth curves in order, and that would be easy to do, because of course it would be the lag phase, then we go to the exponential, and then we go to the stationary, ultimately to the death phase, which is a common sense sort of thing. So that really covers what we need to cover for chapter 6 in our exam. I hope it hasn't been too painful, but anyhow, we'll start on chapter 9 moving forward for Thursday.